Welcome back for part 2 of Meet the Clades, number 1 of 4. So today, this part of the video will be focusing on early reef builders and biomineralized organisms. So we covered a little bit of this during the taphonomy and biomineralization lecture when we were talking about potential biases in the fossil record. But we're going to explore it a little more in depth today and talk about some of these organisms in detail. So here's just a basic timeline of some of the major events. We have the first evidence of life on Earth. That's our prokaryotic critters. We have our first eukaryotes. We have our first animals showing up. Remember, animals are only a small piece of the eukaryotic puzzle. Then we have something called the Ediacaran Biota. That's a group that we'll talk about here in a few minutes. The first widespread biomineralized skeletons, so things with hard parts. That's going to be called the small shelly fauna, coming in right before the Cambrian explosion, where diversity on Earth really takes off. All right, so the origin of animals. The relationships of animals to other precursors is not entirely clear at this point. As you can imagine, delving back hundreds of millions of years into the fossil record can be a little bit difficult, especially when the things that we're looking at in the fossil record are entirely unknown to us today. You know, they look incredibly different. Now, we have many definitive modern groups by the Cambrian. So trilobites belong to Phylum Arthropoda, the same group that belongs, lobsters and insects and other things belong in as well. However, a lot of these groups have possible origins in the pre-Cambrian, the super eon that contains the Hadean, Archean, and Proterozoic that lasts up until 541-ish million years ago. A lot of these critters are called the Ediacaran Biota or the Ediacaran Fauna. They mean the same thing. Don't worry if they're different. I usually go with Ediacaran Fauna. So these are molds in coarse, clastic sediment, usually sandstone. Why would that be strange? Well, take a look at A here. A is this gorgeous critter called Dickinsonia. Absolutely beautiful. Look at that preservation detail. Just perfect. How did that form in sand? Shallow water. Coarse. Doesn't that usually mean high moving or higher energy? If you're remembering your said pet days, you are correct. Absolutely. So the Ediacaran fauna represent a really strange taphonomic window into life before the Cambrian explosion. Now, again, we always have to ask, are these fossils the first animals? Or are they the first fossilized animals? Or hey, are all these things even animals? There's a whole bunch of questions here. What they have in common, though, is that they're soft bodies. They resemble sea pens and jellyfish, other quilted radial organisms. So they're named for a Lagerstätte found in the Ediacara Hills of Australia. This was found by a guide taking his students out on a field trip, and they came across this accidentally. And for a long time, everybody pretty much ignored what he had to say because there's no way that life was around before the Cambrian explosion, right? So unfortunately, fortunately, I guess, for him, he was completely correct. And once people started finding these deposits globally, people started to recognize that, yes, the Cambrian explosion was not the first thing, or not the first evidence of life on Earth as we had originally thought. Now, the Ediacaran Biota, just because they're linked together by a fun name, doesn't mean they're actually linked together because they're closely related. They're linked together because they're preserved similarly, right, in the coarse sandstone. So we're not entirely certain how they're related to each other evolutionarily. But they're coming in right here in the Ediacaran period of the Neo-Proterozoic era of the Proterozoic eon. So the Ediacaran Biota, these are some famous critters here. Dickinsonia and Kimberella. So I know that this one looks more like a dumpling, but I assure you it is a fossil. All right. So these are some of the famous creatures coming out of this. And they're captivating because there's so many mysteries involved with how they lived, who they were related to, all these questions, what happened to them. We really don't know a lot of these answers. This is another, Carnia. This is a frond-shaped fossil. We don't know its evolutionary relationship specifically on this one yet. But some of them got really large. Like, Carnia was actually taller than a 6-foot tall human at some point in time. Dickinsonia also got very large, over 3 feet in length just about. So while I say that they got large, these were also incredibly flat at all times. So don't forget that. We also had trace fossils. Now again, trace fossils don't tell us too much about how many species there were, because remember, one organism can make multiple trace fossils, but it does tell us a little bit about the behaviors. So there was a little bit of burrowing, not a lot. There was a lot of scraping, other things resting traces. So we know that they were doing something. Here's just a lovely diagram showing Dickinsonia here. We have our Carnea here. We have our Cambrian hanging out here. This is by Franz Anthony, an incredibly talented paleo artist. So they required exceptional preservation to fossilize. And what's strange, though, is we have them globally. They're known from every continent except Antarctica at this time, which is really nifty. Here is a map showing all of the possible all of the known locations at this time. Namibia has incredibly beautiful ones, especially with trace fossils. The White Sea in Russia recently uncovered approximately 700 different Dickinsonia specimens. Absolutely wild. Now, there's a lot of hypotheses that have been thrown about with the Ediacaran relationships. I will start this off by saying there is no one correct answer, nor do we know if any of these answers are correct. All right? So some folks, like Adolf Seilacher, a German paleontologist that passed away a couple of years ago, suggested they were an evolutionary dead end. Everything that the Ediacaran represents went extinct. That would be called the Vendobiota. A lot of them probably did go extinct, but we don't think all of them did. Some other folks have suggested maybe they were a type of fungus, lichen, and maybe some of them were living on land. That definitively ignores a lot of the sedimentological characteristics that tell us that we are smack in the middle of water. Almost certainly not that. Maybe some of them are precursors to modern animals. Let's investigate that hypothesis, and specifically, we're going to use Dickinsonia to start. Dickinsonia, after some incredibly well-preserved specimens were found not too long ago, 3 or 4 years ago, I believe, they were found to have cholesterol, which is a compound that can only, or let me rephrase, is only known from animals today. And this signature was found across multiple specimens. So we do think that there is evidence to suggest it might have been an animal. Well, an independent study also found a growth series of Dickinsonia specimens by quite a lot of them, and they were able to understand how it was growing and the process by which it was adding segmented divisions to its body. As you can see here from this chart, you may not know this, and that's okay. You may not know how worms grow, but this is how worms grow. So between the fact that it is growing very similarly to a worm and the fact that it has compounds that only animals have, these are two strong points of evidence that supports the hypothesis that Dickinsonia may be a precursor to modern animals. And in fact, we have decided that it is likely belonging to the phylum Annelida, the worms. There are other examples. For example, Cambrian. Let me go back to Cambrian here for a minute, just so you can see this here. We think that it might be a representative of an early mollusk. However, we don't have the compound steroids to tell us more definitively. You'll notice I did not use the word prove that it was an animal. Again, scientists don't use that phrasing. We support or reject our hypotheses. So let's move into the small shelly fauna, another excellent mystery in science. Here they are. What are they? We're not really sure at this point. So let's talk about what we do know. They're beautiful, right? But that's not really scientifically helpful at this point. They are small, itty-bitty skeletons or pieces of skeletons from the late Cambrian, or late Ediacaran to early Cambrian, primarily the early Cambrian. So the Timonian was a regional name. This is now the Cambrian stage 2. That is the internationally accepted name. It represents the origin maybe of widespread biomineralization. It does coincide with first evidence of burrowing, strong evidence of burrowing. Potentially, could this be an adaptation to predation? Remember the arms race hypothesis, where predators and prey are evolving in this long-lasting dance to try and outlive each other, right? Could this be an example of the arms race? If these are able to burrow and they have harder shells to avoid predation, Could this be what caused the extinction of the Ediacaran fauna? Were they outcompeted? Were they predated? We don't know at this time. That is active research going on. So let's move into the Cambrian. Almost every phyla shows up in the Cambrian or before it. All right? So sponges here, sponges are all the way on down here. Now this tree may have slightly different positions depending on what form you're looking at. We're not going to worry too much about that. But sponges certainly show up before the Cambrian and even before the Ediacaran. And we do have fossil evidence for that. So a lot of animal groups are a lot older than we originally thought. So let's talk about what we see. This is the Cambrian. And if you're looking at these pictures saying, I have no idea what these are, you would be absolutely correct. And I'm so sorry. I just now realized that Wiwaxia here is hidden. At this point, I'm just going to have to say it is too late. We're not stopping now. All right. So check these out on the lectures. This is Opabania. It's got a long proboscis. Very strange looking creature. We have Anomalocaris, otherwise known as Terror of the Deep. So if you would imagine, please participate in this with me. Stick your arms out in front of you and kind of dangle them kind of in front of your face all the way with your arms outstretched. And it would essentially use what are your fingers now to kind of gently scoop things towards its mouth. Predation in the Cambrian was not that exciting. So Wiwaxia here looked like a weird little dish sponge with a whole lot of spikes sticking up off its back. So there's a lot of questions about how these are related to one another. And as you can imagine, look at these fossils. They're gorgeous. These are found in Lagerstätten, specifically the Burgess Shale, which is an incredibly famous Lagerstätte in Canada of the Cambrian explosion era time. Now let's backtrack. The Cambrian explosion was named because we thought it was some of the first life on Earth. Now we know that's not true. We had the Ediacaran before that. Then we were like, okay, well maybe it was the explosion of hard parts. Well, now we know that wasn't necessarily it either. There's other stuff that was evolving at this time that didn't fit with the Ediacaran. And Ediacaran doesn't anymore represent the first animals we know. Now, this is new information. Now, this is an image coming from the Doshanteau Formation. If that sounds familiar, we talked about this in our taphonomy lecture with phosphatization. The embryos that we talked about under phosphatization come from the Doshanteau Formation as well. This is the earliest known animal evidence today. This might change, but today this is what we know. The oldest sponges are coming in nearly 100 million years before the Cambrian explosion. So what you're looking at here are spicules, the skeletonized parts of sponges. So let's talk about these groups. We're going to start with these three groups and we're going to slowly make our way across this lovely, lovely tree here. All right, so we're going to be talking about Porifera, sponges, Ctenophora, the comb jellies, and Cnidaria, the true jellies. Here is a fun comb jelly. This is the bloody belly. Great name, right? So sponges, Phylum Porifera. So first thing you need to know is they make some excellent memes. These are the pore-bearers. That's where they get their name from. All are aquatic. The majority of them live in the ocean. Some have photosynthetic symbionts, so things that interact and live with them, essentially. So a lot of people think sponges are boring, they lack symmetry, they lack tissues that are divided into different organs, but their one fun party trick is that you can put them in a blender, blend them up, pour them back out, and they will put themselves back together. There are very few animals you can do that with. That is a joke. There are no animals you should do that with if the federal regulations committees are listening. Okay. So here are some examples of sponges. There's a couple different groups that we'll talk about. The Demo sponges. These are silicic skeletons. They're marine, brackish, and freshwater. Here is what they look like in the fossil record. They do not make the most beautiful of fossils. A lot of the times they just look like weird little pellets. But there you go. As you can imagine, their fossil forming potential is not great because there's mostly soft tissue here with some harder skeletal parts. Calcareous sponges. These are made out of calcareous material, CaCO3. They are definitely better at making fossils. They almost always have triactine spicules, so meaning in groups of 3, 1, 2, 3, 4, 5, 6. This is one of my very favorite fossils in the whole world. This is Astriospongia. It gets its name because of the star-shaped spicules. Absolutely gorgeous. They need hard substrate to survive. You'll notice that they have to live in shallow marine. We'll talk more about why that is later. We have our glass sponges, which are gorgeous, all right? So this is one of our glass sponges here. This is a modern one, and these are what their spicules can look like. They look more like snowflakes, right? Just absolutely gorgeous. A lot of the times they'll show squared materials here. So in macro, when we're looking at like a structure like this, we'll see them looking kind of squared off like this. Not necessarily when we zoom in. We also have some strictly fossil groups, the stromatoporoids here on the left and the archaeocyathids here on the right. Stromatoporoids are some of our first known reef building organisms, save the stromatolites. They were common in the Paleozoic, but pretty much gone by the Mesozoic extinction event, the KPG. So these are layered. You can see these gorgeous layers. They're identified by mammellans, which are these bumps right here on the top, named after mammals. That's where they get their name from. And then they have the pillars and lamina. The pillars are vertical lines running across it. The lamina are the horizontal lines. We also have the archaeocyathids. For a long time, we really weren't sure where archaeocyathids belonged. We're fairly certain they go with sponges now. These are a relatively unsuccessful group because they go extinct by the end of the Cambrian. Most of the time, you'll hear folks refer to them as a double-walled ice cream cone. I'm sure you can see why. Here's what they look like in life. We have our mammellans, the little bumps, the lamina and pillars. We also have our double-walled ice cream cone. Sponges are porous. They lack symmetry. They don't have specific tissues or organs, so they're pretty simple animals, all right? They do have different cell types, outer layer, inner layer, and middle layer. And so we're not going to worry too much about these cell types, so this is what they look like. They draw in water through their pores, and then it expels out through the top, called the osculum. And this is what that looks like. The water's going in through the pores, and it's coming out the top, the osculum. Super fun, right? Let's watch that GIF one more time. Feel like there should be dramatic music or something. All right, so let's move on our climb through the phylogenetic tree here. The Ctenophora and the Cnidaria. And if you're thinking, wow, those two look like sister groups, you would be absolutely correct. Excellent job. You would not be correct if you said that the sponges are sister group to these two. The sponges are sister group to this entire part of the tree. Excellent. All right, so Cnidaria. We're not going to worry about the ctenophores because they have a really terrible fossil record. Cnidarians are corals, anemones, and jellyfish. Cnidarians have radial symmetry. They also have true tissues, including inner and outer body walls and stinging tentacles called cnidocytes. English is the worst. I do not know why there is a silent C, but it is there in cnidocytes and Cnidaria. My apologies. So cnidocytes, you can also call these the stinging cells. Hopefully this makes sense. Jellyfish, obviously, that's what they're famous for. Corals have them too. Now they do have a nerve network, but they do not have a true nervous system. Now they have one of two adult forms, a polyp, much like a coral, so it doesn't move, or medusa, like a jellyfish. All right. This does mean that corals do have a medusoid phase, and it does mean the jellyfish have a polyp phase, which is really cool. So Cnidaria can be broken up into the scyphosans, the hydrozoans, and the anthozoans. Now, I'm going to be really clear with you on what names you need to know, all right? So really, I want you to know the anthozoans. I want you to remember the name Cnidaria, but spelling isn't super important to me, and your exams are open book, so which means I would hope that you spell at least accurately enough for me to figure out what you're saying. But the scyphosans, the true jellies, have a terrible fossil record. This is one of the few, right? Super cool, but not the best fossil. Hydrozoans are our friends like the Portuguese man o' war, not a true jellyfish, all right? But super pretty and very mean. And then finally, we have the anthozoans, which are going to be our corals. That's the only group we're going to concern ourselves about here. The corals are important fossils, so you're going to need to know these words because you're going to need to talk about them. So first, here's our skeleton. It is called the theca, otherwise known as the cup. Most of them build vertical partitions called septa. So let's look at that. Here are our septa. Gorgeous. Many, but not all, also have tabulae to support the growing organism. So here are the tabulae moving on in. The zooid, which emerges from the top, is a soft body that's not really preserved. Here's your zooid. And tabula aren't seen unless the fossil is damaged. So what we know from the fossil record, taphonomy gives us that window. So some corals are solitary, many are colonial. All right, corallites refer to the individual bits of the overall coral body. Now, most of the time, these corallites are clones of their parents. There's three major groups we're going to talk about. The Rugosans are the first group. Rugosa comes from the Greek word for wrinkled. Hopefully that makes sense. They've got some nice little wrinkles going on. These are both colonial and solitary. The thing that we need to know is that they are Paleozoic only. They kick the bucket at the Permo-Triassic extinction, and they kick it hard, okay? They have I already said that. They have 6 primary septa. However, it's built in stages of 4. So that's something important to know. They'll come in 1, 2, 3, 4, 1, 2, 3, 4. They don't all add at once. They're made out of calcite, and they usually have those horizontal divisions called tabulae. This is what they look like here. So here are our septa. You can see those vertical lines. We can't see the tabulae from this particular angle, but they're there. We can see them here. These are the septa, these horizontal lines. Now we have the tabulate corals. They definitely have tabulae. Here, those tabulae. Again, we can only see this because this is a cutout line drawing. They have weakly developed or no septa whatsoever. They are not solitary in any way, shape, or form. Excuse me, they are only colonial. And again, these are Ordovician Permian only and calcite. They also do not make it past the extinction. I do want to point out that you have 2 Rugosans in your fossil kit. Please go look at them. These are both solitary organisms, and you could notice them by this shape here. Kind of looks like a tornado shape. Many of them are actually named after tornadoes, or the Greek word for tornado, excuse me. So these probably have photosynthetic symbionts, but we're not going to focus on that right now. Now this brings us to the coral group that you're most familiar with, because these are the ones alive today. The Scleractinian corals. These have 6 primary septa, with later septae built in all 6 spaces. So I'll show you what that means here in just a second. They do not have tabulae. They do have septa, obviously. Colonial and solitary forms. You have an example of one of these in your box as well. Please go find it. Many have zooxanthellae, which are photosynthetic organisms that live in the tentacles and mouth. And they are the reason, let me rephrase, they have such a strong relationship now that one cannot live without the other. So that's an important thing. That's part of what's happening with corals today, is that our zooxanthellae are not able to survive, which means the corals are not able to survive either. So here we have our primary septa coming in groups of 6. Here are our secondary septa. Here are our tertiary septa. So that's kind of how it's added. This diagram isn't perfect. Forgive me, I made it. But this shows you pretty much how they're adding on there. So where do coral reefs live? Coral reefs are excellent facies indicators because when we find them in the fossil record, we know a lot about the environment of the area we found them in. We have coral reefs all over Sweden from the Ordovician, which means Sweden was at one point in time a tropical paradise, and plate tectonics really, really probably burns a lot of Swedish people up right now, right? They're just like, ugh, we couldn't stay there? Yeah. So corals with photosynthesizing symbionts must live in clear water in the photic zone, meaning able to get sunlight. This is usually approximately 50 meters to 150 feet deep. Most symbiotic corals prefer warmer water. They also are found on coasts without upwelling. Upwelling brings lots of sediment in. They hate sediment. Coral reefs can be absolutely massive. Here's the Great Barrier Reef, which is over 2,000 kilometers in length. Now, corals aren't the only things that make reefs, though. Not even close. So all of the organisms that we've talked about in the second portion of this lecture video are reef-building organisms, too. So let's start with our first friends that we talked about, the stromatolites. Stromatolites were the first of the reef builders. Those aren't on this chart, but they were there. Remember the layers of biofilms? We have algae. Oh, excuse me. Yeah, we have algae. We have sponges making our reefs. Sponges were incredibly important in the Permian for reef building. Archaeocyathids, which again are a group of sponges, were important in the Cambrian for reef building. Stromatoporoids, the group of sponges, became very prevalent in the Devonian. Bryozoans, a group we'll talk about later, corals here. Bivalves actually built coral reefs. Not coral reefs, they built reefs. We'll talk about rudist bivalves, which were massive. So there's a bunch of different groups that make coral reefs. I'm going to stop saying coral reefs, my goodness. So there's something important we need to understand about reefs. Reefs are incredibly important to our ecosystem. We know that today, right? All the scientists are worried about climate change affecting coral reefs. Coral reefs are in major decline, and we are worried because they hold a massive amount of our biodiversity, and their illness could be what brings down a lot of our ocean life. But what we should be concerned about too is that anytime we have an extinction or reef gaps where we don't have many reefs, we actually have a mass extinction. So where we lose our Rugosa and tabulate corals, we have an extinction. Where we lose a lot of our bryozoan and our other sponge stromatoporoids, we see large gaps in our reef, in our biodiversity. So what we can see here is that reefs are incredibly important, and when we lose them, they oftentimes seem to coincide with our mass extinctions. So the reefs are under threat today. That's not a surprise. This is Lizard Island in the Great Barrier Reef from 2010. Here it is in 2016. This is a 6-year, less than 7 years later, 2016. We see that in Lizard Island, the reefs are dying in droves. See the color differences? This means that the corals are being bleached, which is synonymous essentially with their death. Once the zooxanthellae are dead, so is the actual coral here. So what's happening? Rising temperatures, decreasing pH, so making the water more acidic, more pollution in the water, creating more pollutants, sedimentation type things that are irritating the corals. And this is happening from increased carbon dioxide and increased runoff into waters. There's certain types of sunscreen that is causing the death of corals as well. Make sure you check that out next time you go to the beach. And this is happening worldwide. We estimate that over 90% of our coral reefs will likely be dead in the next couple of 100 years. So this is something that we really need to be paying attention to, and why paleontology is so important. We can use the fossil record of reefs in the past to understand what we might be looking towards and to help us find solutions. Because though it does seem bleak, there are solutions to halting climate change or even lessening some of its effects. So be paying attention to that in the future. All right, folks, I will see you again for the Meet the Clades Part 2.